Mitosis is the type of cell division that creates all our body's cells besides sperm and egg cells. In mitosis, a parent cell creates two daughter cells simply by dividing. Each daughter cell, however, must have its own nucleus with a complete set of chromosomes. The extra copy of chromosomes were created through DNA replication during the S phase of interphase in the cell cycle. And in the process of mitosis, a nucleus will dissolve and two new nuclei will be created. So that process of actually dividing the nucleus and the chromosomes held within it is mitosis. As you can see here, mitosis in an onion cell, you see the chromosomes having separated the darkly blue-stained regions. Cytokinesis is the term for the actual division of the rest of the cytoplasm and the cell membrane itself, and that usually starts during the last phase of mitosis and completes after mitosis, as you can see here with the protist completing cytokinesis. Now, before we can start talking about the phases of mitosis, there will be an important structure called the spindle that's created. The spindle refers to the set of protein filaments that will help guide the separation of the chromosomes when they occur. Some things to be aware of: the centrosomes are the two opposite poles of the cell that will be the organizing center for what are the spindle fibers that reach out from the centrosomes across the length of the cell, extend towards the center. Now these are just microtubules, protein filaments, and the chromatids will attach via their centromeres to those spindle fibers and ride down along them to separate from each other and to head to either pole of the cell. So there are four phases of mitosis, as you can see up here at the top: prophase, metaphase, anaphase, and telophase. We'll start with prophase, the first of these four phases. Now, there are a few things to keep track of as we go through the phase: the status of the chromosomes, the nucleus, and the spindle. So first we see in prophase that the chromatin, the loose form of our chromosomes, is condensing into chromatids, and you can see that quite nicely here, where they will start darkly staining and being very solid form. In our drawing over here, we will draw them as that characteristic four-armed structure, or that X shape, representing the sister chromatids joined by a centromere at the middle. And we also see that the nuclear envelope is dissolving. That nucleus must go away in order to separate the chromosomes from each other. And also we see the beginning of the spindle formation. The centrosomes will organize and start to grow their spindle fibers, reaching out to each other. In metaphase, the second phase of mitosis, we see the characteristic lining up of the chromosomes at what's called the spindle equator, simply the middle of the spindle. And we can see that nicely here, where those chromosome centromeres will attach to the spindle. The nucleus, as we can see, is gone, and the spindle fibers are fully formed and attached to the centromeres. In the actual picture here, you can see those chromosomes lined up down the middle with the spindle in pink reaching across the cell, and they're all those centromeres are attached right down the middle of the spindle equator. Now they're lined up down the middle in preparation for the next phase. Anaphase. Anaphase is characterized by the sister chromatids separating to opposite poles, you can see here. So recall that each of those pairs of sister chromatids were exact copies of each other. So one copy goes to one side of the cell and the other copy goes to the other side. And in that way, we can be assured that the eventual cell that forms here will have one set of chromosomes, and the cell that forms here will have exactly the identical set of chromosomes. So those sister chromatids separate. That marks anaphase. The nucleus is still gone in this phase. And you also see that the spindle will begin to disassemble. We'll start to immediately have the spindle fibers retract back towards either side of the cell as soon as the sister chromatids have been pulled apart. In the final phase of mitosis, telophase, we see that the chromosomes are loosening back into that chromatin form. Remember, chromatin is a relatively loose form of our chromosomes, and you can see that here in these two actual slides of mitosis where the chromosomes are still relatively condensed, and then you see that they loosen up into that chromatin form here towards the end of telophase. The nuclei will begin to form around each of those chromosomes. Now that's hard to visualize here, but there is starting to become a nuclear envelope around those chromosomes here. And of course I've designated that with this dotted line representing the nuclear envelope here. Now also the spindle will continue to dissolve. Again, it's highly variable among cells and species, just the extent of the spindle disappearance in this phase. But we would normally draw them as simply retracting back to their very centrosome nature. And also we start to see the beginning of cytokinesis, the division of the cell. Now this is going to be different for an animal versus a plant cell, and what we have depicted here is an animal cell. So that final phase, cytokinesis, actual division of the cytoplasm. And like we said in the last slide, this happens during telophase. It tends to begin during that phase. And in animal cells, we form what's called a cleavage furrow, this gap or this pinched sort of area here where the two cells are separating. And that's created by a contractile ring of actin filaments. In this slide you can see the actin as highlighted in red here, or stained red. Now that's just a protein filament that's like a string tied around a balloon, slowly tightening, forming that cleavage furrow until finally those two cells separate entirely. In plant cells it's a bit of a different process. Instead of forming a cleavage furrow, because plant cells have a rigid cell wall completely around them, those two new cells have to form what's called a cell plate between the two new nuclei. Now that cell plate is simply a new cell membrane formed between the two new daughter cells. And as they deposit phospholipids at the center, eventually an entirely new cell will form here and a new cell here, and then a cell wall will form between them after that plasma membrane has been laid down.